Let's start. Welcome everyone to today's Monash Advanced Microimaging webinar. Before I start or having the pleasure to introduce our speaker today, Elisabeth Kugler, I would like to acknowledge the people of the Inkulian nations on whose land Monash University is located and pay our respects to elders past, present, and emerging. Today is Elisabeth Kugler, whose journey as a scientist started in Austria, where she graduated from her studies with I would say, probably short overlay in Germany. She then continued her journey in the UK, where she did her PhD at the University of Sheffield, where she used a state-of-the-art light sheet fluorescence microscopy to visualize the vasculature in 3D, in vivo, and for prolonged periods of time. After her PhD, Elisabeth did her postdoc at UCL as a research fellow to build then image-based models to describe neurovascular development in the zebrafish retina. Today, she is still working at UCSL as an associate fellow, but very exciting and interesting, she is also the founder and director of a company that basically combines arts and science, and maybe we will also hear and see more of that. The company is called Geeks, Art for Geeks. Elisabeth has won a lot of awards throughout her career. A few to mention is that she was awarded an Ian Peake Award for Research Excellence, a career development bridging a fund from, if I pronounce it correctly, Insignio, Institute for In Silico Medicine, and an honorable mention doctoral research award in natural and life science. She also is demonstrating a great leadership capacity. She is, again, founder and chair of a departmental student society and member of the faculty PGR society. I don't know what PGR is. Maybe you can.
Postgraduate researchers.
Perfect. And there was one more to say. Yeah. Very interesting as well. She also acts as a faculty peer mentor and student representative at the medical school Athena Swan, which is a scientific women's academy network, which we also have here in Australia. So again, it's our great pleasure to have you here, and I'm very excited to hear your talk about how to quantify a zebrafish research software for image-driven biomedical studies. All yours, Elisabeth.
Thank you so much, and thank you for this really kind introduction. I'm just trying to share the screen. If you can just let me know in a second if that all works. So hopefully you see the screen now.
Yes.
Yes. Okay, great. Thank you so much. Just move you around. Okay. So thank you so much again for this really kind introduction and for having me here. I'm actually in the UK, so it's really early for me. So good morning, everyone, and huge thanks for joining in. So for those of you who don't really know kind of what a biomedical image analyst is, I thought I put in a slide of really saying we are kind of trying to bridge knowledge. We're trying to translate images into knowledge. So we normally get 3D microscopy data and then translate them by writing code or something into and to derive some numbers, such as the number of cells, the volume, or for example, their intensity. And this really kind of this little arrow is actually a huge problem for a lot of researchers. And we see this when we talk with people, but also when we ask, for example, here in this questionnaire from Twitter from last year, and if we say, if you work with microscopy data, what is your biggest challenge? And actually 55% of these 672 answers say it is data analysis. So it is really a huge issue at the moment. And we're really trying to find ways to translate all the data that we can get now into meaningful numbers. And so today I thought, I don't really want to talk about, the maths and the algorithms and stuff, but actually talk about, a little bit behind the scenes and the things that I wish that I'd known when I was starting with microscopy and data analysis. And so it's a really kind of more high level and overview talk, really. And to do this, I will be talking about 4 different topics. The first one is I will be talking about zebrafish as a model. And I'm sorry if you already know about them, but I always think it's really important to make sure that we all are on the same page, especially because really most of my research has evolved around zebrafish. And for today's talk, I will be just talking about zebrafish data. But obviously everything that we developed in terms of data analysis can be also applied to any other models such as mice, human, and other tissues. Okay. And so after I will introduce the Zebrafish, I will be talking about imaging and understanding, and I will be talking quite a lot about that because the better our data, the better our data analysis approaches will be. And I don't need to tell you that as microscopists, but I always think it's really important to look at it from the angle of biomedical image analysis. So we kind of come in later, but we also kind of, have to have this conversation. And then I will be talking about pre-processing and segmentation. So this is where we come in with actually improving the data and then at the end quantifying any meaningful results. So now I will be introducing Zebrafish for a little bit, and again, I'm sorry if you already know about them. But this is what an adult looks like. So they're about 2 to 3 centimeters in size, and that's the size of a matchstick. Their stripes would give them their names, and this is really a very hugely emerging model organism for many biomedical studies. But most of the research doesn't actually happen in adults, but it happens in embryos, because the embryos develop outside their mother, so we can very easily access them, and they develop very, very quickly. So within 24 hours, they form a basic organism where the heart is beating and the blood is flowing. And within the first 5 days of development, really, most of the processes that we as developmental biologists are interested in are already happening. So we can study a lot of things in really very, very short periods of time. And that makes it a great model if you want to answer questions rather quickly. But also, even though they're fish, they are more similar to us than we think. They have organs such as brain, heart, liver, muscles, and many more. But also, we share a lot of genes with them. So in general, we say about 70% of genes. But if we look at disease-associated genes, this actually rises higher to 83%. And this is also important if we look at it from the perspective of actually genetic manipulations and disease modeling, because what we can now do is actually introduce patient-specific mutations and make these fish subject to different assays, such as studying how a certain mutation impacts generation or wound healing, or for example, how this responds to chemical screens. And so particularly here for chemical screens, there's a lot of pharmaceutical companies that actually invest a lot of money in trying to link patient-specific manipulations and mutations to chemical screens, because this is a huge avenue for patient-specific therapeutics. But the thing that fascinates me still the most is really kind of the imaging side of things, right? And so here another aspect makes fish a very great model, and that is that the embryos are actually transparent. So this is what they're looking like. So if the kind of comparison of the matchstick for the adults holds up, then the embryos are smaller than the tip of the matchstick. And so they are here kind of just blown up, and here you see a single cell stage, and this is a 4-day-old fish. And I hope you can appreciate, so here's the eye, the brain, and the trunk, that they're actually almost transparent. So you can look literally right through them and into their brain without any interference, without any fur, skin, or any kind of alterations. And this becomes obviously hugely potent if we combine this with the fact that we can introduce transgenic reporter lines. So we can visualize tissue in living samples without actually any interference. And this is what you see here, where we look at the vasculature in this very same fish. And so we can study, for example, how these blood vessels develop over time without actually having to do any kind of invasive procedures with these fish. This is obviously still limited to the embryos, so the adults are non-transparent, which is still a little bit of a problem. So there are kind of 2 avenues for the adults that are currently mainly followed. The first one is tissue clearing, so fixing the sample, performing tissue clearing and refractive index matching, and then being able to image them in 3 dimensions, or introducing really genetic alterations that inhibit this pigment formation from happening. So these will stay transparent throughout their adulthood. So you can, for example, take away these blue and white and gold pigments and make these fish really more transparent. And what you see here in this case is a beautiful study from 2021, where you look at the top of the brain. Here would be the nose, the right eye, the left eye, and here's the brain. And you see that the brain is like pinkish, but actually the vessels that this group studied are lymphatic vessels. And they can study this in vivo without any interference. And it's a beautiful study. If you haven't read it, definitely have a look at that one, because it's like really pushing the boundaries of what we can do with zebrafish. And so these are really obviously a lot of different characteristics which make them a fantastic model. But for us today, the obviously kind of important point is that they are a great model for any imaging and then data analysis. And so this is now what I'm going to switch to, namely, what are the different techniques that we're using? And again, I don't want to preach to the choir, but I thought, it's interesting to just look at it from the perspective of, fish. What can we do in terms of imaging? And so here we really try and cross different spatial and time resolutions. And so on the left, we have large periods of time, and on the right, we have high resolution. So on the left, we have techniques from the medical field, such as MRI and tomography, or for example, X-ray. As you can imagine, putting a fish into an MRI is rather challenging. So it's not something that a lot of people do, but it is definitely something that is becoming more and more important, because this is obviously data that is very similar to what we can do in human patients. And so if you look at it from the perspective of neurodegeneration, God, long word, Alzheimer's, or for example, aging, that is really important and a very, very great technique that is becoming really more and more applied in longitudinal studies, not just for zebrafish, but obviously also for other model organisms. But the things that are probably the most used across the field are really confocal and light sheet microscopy. And again, I'm sorry if it's just repetitive, but for confocal, what we have is a focused laser light. And that's important because it penetrates into the sample, leading to a whole volume illumination. So it's not very specific, and you actually have a lot of kind of out of focus illumination. And the detection is based on a pinhole, so you kind of scan through the sample. Whereas in light sheet, we use a sheet of light that illuminates a whole selective plane at once. So that's a whole focal plane being illuminated at this very same time. And this whole plane is acquired by an uncoupled detection objective, which is what you can see here, which is based on a whole field detection setup, so to speak. And so light sheet has become hugely important if we think about the fact that this reduces phototoxicity, it's very, very fast, but also because the beam paths are not really, interacting with each other as such, you can really penetrate into the samples very, very quickly. And so this means that light sheet is really important for long-term data and large field of view. And you see this here in this video where we look at the vasculature from 2 to 3 days. So this is 24 hours, every kind of 10 to 15 minutes. And we look at the blood vessels in a 2-day-old fish. So what you see is actually the side view. And here would be the brain, the eye, the heart. This is the egg yolk. Here's kind of the fin forming. And here's the trunk. And it's obviously 3D data, but just shown as a 2D projection just for kind of reference. But what you already see is actually that this field of view is quite large. It's 1 millimeter, which is, if you think about microscopy, quite a large field of view. And also that actually the fish develops completely normally. So there is no photobleaching, no kind of interference. And this is what makes light sheet really so, so beautiful, because we can get these type of data and study developmental processes really over hours and days without being invasive to the samples. So this is kind of, the standard application of light sheet really. Whereas in confocal, we look at a very different time and spatial scale. And we see this in this example, where we look at a region of the eye. So specifically, this would be this region here, just kind of swapped on top, so to speak. And we're looking at 2 different cell types. In green, we see a type of neurons, and in magenta, we see a type of support cells called Müller-Glier cells. And this time lapse is about 4 hours. And what you see is actually that these green cells are migrating, interacting with each other, and for example, dividing. So you see cell divisions happening here. And so confocal is really something that we use to study tissue level dynamics and cell to cell interactions. And again, it's a very different space and time continuum, basically what we look at in these two. But confocal has also been pushed a little bit now with AiryScan, because while confocal is limited to the diffraction, in AiryScan we use combinations of detectors that allow you to push beyond the diffraction limit. So that means that normally we have 200 by 200 by 500 nanometers resolution, so this is X, Y, and Z. And in AiryScan, we can get 120 by 120 by 350 nanometer. And this is great if we look at very fine cellular details, such as this one here, where you look at cellular protrusions that are trying to look at other neurons. And so specifically what you see is actually one of these magenta cells, but acquired with AiryScan in a slightly different visualization. But you can already see that we can now show they have different subdomains, and all these very fine protrusions are visualized just with this AiryScan microscopy. If you don't believe me that there's a huge difference, you see this also in this one. So here's confocal and AiryScan, so it's the same image, but acquired with confocal and then with AiryScan. And not only do you see that this one is brighter because you have a higher signal to noise ratio, but also you see more detail, such as, for example, in these cells here, or when you look at these positions here. And this is because in AiryScan, you generally have this higher resolution and higher signal to noise ratio. And so this is just one example, but this is really kind of now where the field is really going, trying to push away from standard confocal to more resolved data acquisitions. And then lastly, on the right side is obviously electron microscopy that every one of you knows. And this is obviously a single time point. So these animals are not alive anymore, whereas for all of these techniques, the animals are generally most of the time still alive. And here we obviously get the advantage of having a very large or very high resolution. So for example, here, this is a blood vessel that you're looking at. So this blood vessel is about 10 micrometer in diameter. And you see that we can get a resolution of about 4 by 4 by 60 nanometer. And again, this is not something that we use as a standard in all the fields, but really we mainly focus around light sheet confocal and AiryScan data, and a lot of fluorescence and in vivo imaging. And for today's talk, I mainly will be talking about light sheet microscopy, although there will be a little bit about confocal and a little bit about AiryScan later on as well. But for now, I want to switch to kind of image understanding, because we all know this triangle of compromise, but I always think it's also important to look at it a little bit more in a wider scale in terms of, what does this mean for example, sample health, or also the data that we look at. And as an image analyst, I often see the same problems over and over again. And so I thought I just want to very briefly highlight them today and kind of, share that they are still a huge problem, even though a lot of you obviously know already that we need to look at these things. So for example, for confocal type imaging, we have two main issues. So the main thing that I often see is a problem with sampling frequency. And so this can be obviously in different dimensions, but I'm just showing you a sampling frequency problem in the z-axis in this case. So what you see is the same image in X and Y on top of here, and then we look at the stack from the side, so we basically just reslice it, and you see the individual slices. In this left one, we acquire them with 1 micrometer intervals, whereas on the right, we acquire them with 0.19 microns, so it's about a factor of 5 difference. And I hope that just by looking at these two images, especially in these regions down here, you can appreciate that there's a huge difference in detail that we can acquire and just really gather by this increase of sampling frequency. And here we really lose all this detail, and we wouldn't be able to quantify any of these fine details. And this is something that I really over and over see, and this is especially something that before you acquire a whole data set, it's always worth going to your data analyst or your colleague and say, is this the right sampling frequency? And, what is it that we need to improve? So this is really the main problem that I see over and over again. The other thing is signal decay, where we again in X and Y have a very homogeneous signal distribution, but obviously because it's a focused laser light in confocal, if you look again at the set stack from the side, so here would be the top of the tissue and here's the bottom of the tissue, we have a very rapid signal decay. So the first micrometers, we have really nice signal, and very suddenly we have this drop off, and we see this here in the quantification as well. And this is obviously because the focused laser line penetrates into the tissue, and within a few micrometers, you have a lot of interaction of light with matter and just scatters away, and there's no signal left basically. And this is a huge problem if you want to quantify something here and the same thing at the end of the stack, because obviously the data itself is not really the same. So this is something that, again, I see over and over again. On the side of light sheet, the data can obviously also be wrongly sampled, but for example, in light sheet, the main two problems that I see are calibration issues and shadowing. So for calibration, I mean calibration of the light sheet with each other, so if there are multiple ones, but also the light sheet actually, so the focal plane, so to speak, of the light sheet to the detection objective. Because often there is, people go to the microscope, it's really early and they're stressed and they are like, yeah, it will be fine. But actually later on, when you look at the data, you see that actually the calibration was off just by a little bit, but it makes a huge difference. And this is what we see, for example, here, where on the right we see the structure of the eye and this vessel as well as the fin, but on the left we can't see any of that. Same goes for shadowing, which is a problem where we have laser light interfering with, for example, pigments. And you see that this brain, which is meant to be completely green, has some regions which are darker, like for example here this is meant to be green, and some which are really, really bright. And this is something, again, this can be done either by, for example, pivoting the light sheet or by actually bleaching the sample. So again, there are multiple approaches that you can address this with. But often, if people say to me they want to quantify things and I get data like this, I'm like, are you sure? Is this, the best data that we can get? And so this is really just important considerations. But I think also the thing that struck me the most in the last few years is actually rethinking the way how we visualize certain tissues from a biology point of view. And so I just want to share some examples with you. So the first one is when we actually look at, for example, microglia cells. So these are the support cells of the eye. We have a standard transgenic reporter line. So that has a M-cherry in the cytosol. And this is normally what we see. But actually, what we found is that if we produce a new marker that actually has a GFP tagged to the membrane, we suddenly can really change the detail that we can see. So in this case, it's a DNA injection, and this is why it's only single cells. But I promise you it's the same, and you see this also here in this overlap. These are the same cells, but you see that there's this huge difference in quality just by looking at them, right? And the same goes then also for the data analysis, because obviously the data on the left are so much more clear, right? So you see all these details of honeycombing structures, you see the cell bodies, you see protrusions, and it's continuous cells. Whereas on the right, you basically just see the cell bodies, right? And so I think this is really important to always just go back and say, is this the best way that we can actually look at our data? Because this makes a huge difference in terms of what we see, what we quantify, and really the very intricate detail that we can analyze these data with. And so this is for me kind of the epiphany of 2023, let's say it like that. And so now that I've shared with you a lot about Zebrafish and about imaging, I now come actually to the pre-processing. So I think about halfway through, and this is now the part that I'm really interested in what do we actually do with the data, and what does this mean? And for this I will be talking about light sheet data, particularly about light sheet data of the brain vasculature. So this was my PhD project at the University of Sheffield, where we wanted to quantify how these blood vessels develop and form, and what are the factors that impact that. And so just give you a quick orientation, this is the bright field, so left eye, right eye, and here would be the brain, and this is the vasculature as a maximum intensity projection. And we look at the posterior part, so we don't look at the gills or the heart, which would be kind of frontally of the fish, but we look at this posterior part of the head. And this is what it looks like in 3D rendering. And so what you can already see is that actually, when we look at scale bar, that this width of the brain from the left eye to the right eye is about 600 micrometers. So this is about the width of a credit card, which is in microscopy terms, again, actually quite large. And this is, as we know in light sheet, also often a problem that actually the data load is very, very challenging. So it very quickly goes to gigabyte and terabyte in terms of data load. But also a thing that you probably can't see as such, but kind of think about in the theory, is actually that blood vessels are a very scarce data point. So if you think about a brain, it's a very solid object, right? And it's very dense. But blood vessels are actually more like a tree branches, so they're very scarce. And if I look at it from a data point of view, what I see is something that is biased to the background. So there's more background than actually data in the images. And that is something to just think about, because this allows you to maybe optimize how you work with this data load, but also think about what you actually do with the data. So that's already 2 points, very large data, but also very scarce data. But then moving on from this, we typically spend a lot of time with understanding data. So this is really sitting down and scrolling through the stacks, looking at them from 3 dimensions and trying to understand what we're looking at. In the case of blood vessels, when we go through the stacks, these are individual planes that you can see here. When we look at the blood vessels, this is what we see, because we visualize the endothelial cells that actually outline the vascular walls. So it's basically the outside of the blood vessel that you see rather than the inside. And this means, from a computational point of view, that we get something like this donut shape. And when we analyze it, it's a so-called double peak intensity distribution. So this is just plotting the intensity, right? But this is different to what we would see in unluminized vessels, or vessels that are collapsed, or for example, vessels where we inject dextran or where we perform microangiography, where we have a really very nice Gaussian distribution. It's just like a single peak. And as a matter of fact, as always in biology, normally you have both cases in the same data. And so this is challenging because you need to think about, can you standardize them to one way, or do you analyze these ones different from these ones, or how do you approach this, right? And so this is just a very general kind of consideration that I will come back to later. But again, the same here also goes for understanding, the best way to visualize your data. And in this case, we just have 3 different transgenics, which are all meant to show the blood vessels. But as you see, this one on the left shows significantly more like specific signal to the blood vessels. Whereas here, these 2 green ones, you see that there's a lot of like signal in the eyes that you can't see in here. There's a lot of signal like here in the otic vesicles, which are like the ear of the fish, and also more unspecific background. And this, together with like signal to noise ratio measurements and, plotting these cross-sectional intensity distributions, really led us to converting to just using this transgenic and optimizing all data analysis for one specific visualization approach. Again, this leads to, is this the best way that I can look at my data? And so this is kind of what we normally do. We really go through, characterize the data that we see and trying to optimize the data acquisition. But also we think about data variability a lot, and this is obviously within images, so such as this signal decay when you go into the stack, but also is, for example, data acquisition standardized in a lab? Because often I get data that are meant to be the same and they're completely different. They can be different in image quality, they can be different in, the position where we look at in the sample, they can be different really in the way they're constructed as such. And all of this can really lead to problems further down the line. And so I always suggest to kind of have open conversations in the lab and say, okay, how do you get these data? And really make sure that in the lab there's at least a very coherent, anatomical location, laser power, things like that are really, really important if we want to standardize data acquisition. And on the other hand, the same goes for understanding our kind of side, the code and data analysis side of view, because obviously we could work with Fiji, Python, MATLAB, Napari. There's so much, and all of them are really different. And so it's really important to early on speak with researchers and say, what is it that works for you, and what is the best approach for any data analysis? And even within this, it is really important to test how different data respond to, for example, different filters. And I just want to show you one example from my PhD again, where we wanted to filter basically our blood vessels. And so the idea was that we improve data quality within the blood vessel and suppress any signal that is outside of the blood vessel. And there are various different filters, but I'm just showing 3 examples. And in theory, at least those 2 are meant to show more or less the same. But already, as you see, A, they're not able to really work with this type of data, but also B, that they're very, very different to each other, even though they're meant to be very similar. This one, on the other hand, is working really well for our data, which was very surprising. And so we kind of just moved on using this one, and then we'll talk about this in a second a little bit more. But this is really just to highlight that code implementations can be very different, and you shouldn't just blindly trust them. Always make sure that you test it, and then that you look at what does it do to your specific data. So that is really, really important. And the same goes with, for example, any common filters which are normally used to, for example, remove any background or remove any kind of scattering, etc., which I just call like general filters or like common approaches here, which are, for example, median filter or like a rolling ball, which a lot of people use to improve your signal quality. But the important thing is that these are unspecific filters, so they're not really tailored to your data. So you could apply them to a blood vessel, to a brain, to cell nuclei. These filters don't really care, right? But there are filters that do care, and this is important if you have like shape-specific structures, such as, for example, cell nuclei that are round, or, for example, blood vessels that are tube. And I'll be talking now a little bit more about this tubular filter, because this worked really, really well for our data, because it assumes that our vessels are a tube. And so I won't be talking about maths, but what it does is it improves all the signal that is along and inside the vessels, and it tries to suppress everything that is outside. And this actually comes from the medical field. but works surprisingly well in our fluorescence microscopy data. So this is medical data, and this is just one example of our data where you see you have a significant improvement of the signal. So this is before, this is after. It can deal with little speckles, such as shown here, and it can suppress really the background really, really well. And so this is where a shape-specific filter is really great. And this is especially the case when we look at this kind of case of this donut shape where we have these luminized vessels versus these unluminized vessels. Because what this filter does, so it's called Sato enhancement or tubular filter, is it actually, like I said, improves everything along and inside the vessel. And so it takes both luminized and unluminized vessels as an input, but actually outputs everything as filled blood vessels. And so you no longer have to deal with two different situations, but actually make sure that all the vessels are the same to deal with. And that is a huge thing if you think about it computationally, because suddenly you don't have to think about 2 points, but actually you now can work with data as one kind of continuous structure, so to speak. Once you have this, and obviously we have done loads of kind of analysis on this, we then move to segmentation. And this is particularly at the moment a lot in the news, especially with like machine learning. So we'll talk about this in a second. But for now, I just want to highlight again that there are two main approaches to this. The first one is semantic, where you just try and find like a foreground and a background, such as persons and, background, or instance, where you try and find individual people. Instance segmentation makes sense for nuclei, such as nuclei 1, 2, 3, etc. But for blood vessels, it doesn't make sense because blood vessels are normally one coherent vascular tree. So they're all connected to each other. And so in general, what we do for blood vessels is that we only use a semantic segmentation. So we say, is it a vessel or not? That's all that we do. And here again, there are various different approaches. And what we found is actually that for light sheet data, for the data that we look at, intensity-based segmentation works really well. So this is basically just like an auto thresholding that we do here. And this wasn't really surprising, but what kind of was the learning point for me in looking at all of this was that when we looked at more sophisticated things such as level set segmentation, that they really struggle with our data, not only because it's fluorescence microscopy and very different kind of type of data for what it was kind of originally developed, but also because especially in light sheet, the data are just really, really large. And so this runs for a week and then it crashes and then it runs again. And then, so this is very tedious work. Or like, yeah, I tried it a few times. I was frustrated. I gave up. I moved on. So, and the same for many other processes, obviously. And I think that's a very important thing as well. You need to find a sweet spot of what makes sense to have in terms of time investment. Does it make sense for one fish to run a segmentation for a week? Probably not. So you want to switch to something that is like this, that runs for 2 minutes and it's done. And that is really, again, something that is important to consider, time investment for all of this. And I mentioned machine learning very briefly because we have actually looked at machine learning for segmenting the vasculature. And this was a very kind of brief study where we just looked at different architectures and whether they would be able to work with our images. And I have to say, I did doubt it at the start. I thought it wouldn't be able to work with our data because they're so different to anything that is, that we normally kind of segment with this type of machine learning. But actually, it did work really well. And what you see here is in green, our segmentation, and in magenta, we see a UNet-based segmentation, and in white is basically the overlap. And I hope you can appreciate that there might be some differences, right? But the overall segmentation works really well. And this is important because normally machine learning is trained on a slice-by-slice basis, so it doesn't work in the 3D space that the data actually acquired in, but works on slice-by-slices. But also because obviously if you go through the stack in 3 dimensions, the kind of data structure, like where are the vessels, how bright are they, etc., really varies. And still the machine learning is able to pick up on this and still delivers a very, very good segmentation. And so with a little bit of tweaking, etc., I think you can get a very good segmentation that very quickly works on your data. Let's say it like that. So if you have time and are interested, I think this is definitely a very good way to invest time in to work here with machine learning. And so that's probably also the point that most people kind of, use machine learning for actually data segmentation. And so now for the last few minutes, I want to talk about, the quantification. What do we actually do with these data once they are segmented. And so normally what we do is we try and quantify different features. But to start, or before we start, we also have like a step of validation. And that's very important because obviously data can be very different. And if you have never worked with a data set, it's very difficult to be kind of objective and say like, you know, is it really the best segmentation? And so normally what we do is we compare different approaches against each other. And this is exactly what you see here, where on the left we have a vasculature. So this is the original maximum intensity projection, and this is a segmentation after general filters. So this is just a median filter and a rolling ball segmentation, followed by Otsu-based thresholding. And it looks very decent, right? On the right hand, we have this Sato enhancement, so this tubular filtering, and then apply an Otsu-based thresholding. And again, if you compare those two, there might be some points where you say, okay, this looks better here, this one looks better here, etc. But you can't really quantitatively assess, you know, which one is better in just by looking at these. And so you need something to address this and test this. And we looked at different data sets, but today I just want to show you one example where we looked at images, again at 4 days, where we image the samples, we take them out of the microscope, we open the heart cavity and let the blood leak out. And basically, that means that the blood vessels collapse because obviously the blood kind of, you know, drains out and the vessels start to constrict. And so our assumption was that, in theory, this is more vascular volume and this one has less vascular volume because the vessels are constricted. And when we quantify the volume in these two using those two techniques, we find the following. So when we do the general filtering, so we use this one and measure the vascular volume, what we see is that not only is this not able to find that there is a significant difference, but also this one has a huge coefficient of variation. And biologically, this is not what we expect. We don't expect, you know, a fish to have 3 times the vascular volume than its kind of siblings. You would assume that there's a way lower coefficient of variation. And we actually do find this when we do the same analysis, but just with this segmentation approach, where we now start to see that there is a significant difference between them. And also we now start to see that there is such a low coefficient of variation, which is about 15%, which is exactly what we would expect from a biological point of view. And so this is just one example of how you would assess, you know, is my segmentation really the best that it can be? And so obviously from there on, we only used this one because we were very confident that this one is the better segmentation for our data. But it's just one example, right? So you can do many more things, and I'm happy to talk about this if anyone is interested on how to validate your segmentation. But actually, normally we then, you know, move on to say, okay, now what do we quantify? And this is something that I talk a lot with different scientists, because a lot of the time people come and they're like, you know, want to quantify everything. And it's, you know, in theory, we can quantify everything, but there's a question of like data overload and what makes sense and, you know, what are sensible data. And so I think you kind of need to look at it from a different spectrum of like high level to really small scale data analysis. And so I just picked 2 graphs here and I will talk about them in a second, but basically they will show this breadth of, you know, very large scale kind of analysis versus really fine scale analysis. And we can measure anything in between really, but it's important to find this right spot. So as an example here on the left, what we have is data from the brain vasculature, so these light sheet data, where we measured the vascular volume, or for example, the branching points. So for each brain, we get 1 point. We say, for example, there are 400 branches in one particular brain. And we measure 8 different parameters, and all of them are on this very high level analysis. But still, when we do this and apply this to different drug treatments, for example, and morpholino-based approaches, we can pick out that there are a lot of significant differences, even when we just look at this really high-level data analysis. And this makes sense for this type of data. So for example, here we can inhibit vascular growth with this VEGF inhibitor. And we see in magenta that there's a decrease of most of the parameters. Whereas if we do the opposite with NOx signaling, we see that there's an increase of most of the parameters. So this is a very nice kind of control that we are sure that we can measure in both directions, increase and decrease, right? But this is just one, you know, really high scale measurement. But this is often what we do in these really large scale data, because it wouldn't make sense to get hundreds of data points for one particular brain. But we can also go the completely other way, which is really subcellular detail. And this is what we see here. So we see an individual molecular cell, so these cells in the retina that I showed you before, which have a top part, and then they have a cell body, they have a region that connects with neurons, and then they have an end foot that ensheaths the blood vessels in the retina. And what you see in color codes is actually different orientations. So in red, you see that they are aligned upwards, whereas, for example, in blue and green, they are having an alignment which is lateral. And so this is subcellular, right? So these are just a few micrometer directions. And we can take this approach and actually apply it to three-dimensional data, which is what you see here. So this is development from about 2.5 days to 3 days to 4 days. And here we work together with Stefania Marcotti, who developed this approach to actually, in basically supervoxels or subregions, measure the alignment. And this is what you see plotted here, but also here in color. And I hope you can appreciate that we see this red, which is this, upwards alignment, but also over time, what we see is actually an increase in this kind of green section, which is actually these protrusions starting to form and really mature, and they start to grow in thickness. So you see that the thickness of this region really increases, but also, and very importantly, that actually these protrusions, there's more and more protrusions that are horizontally aligned. So over time, we have this subcellular rearrangement, allowing these cells to mature over time. And so originally we started with 3D data, then we plotted as a 2D here, and then we plotted again as 1D vector to get this kind of data. And this is just showing basically the same, but now as a quantification, where you look at 60, 72 and 96 hours post-fertilization. And now again here, so this region would be the quantification of this region. And you see that over time, again, you see this increase. So darker blue is now this increase in this region, but also that actually over time we see these two subdomains forming. And you see this also like in this image here, which is again like this more sub-layer maturation, so to speak, of this layer. And this is really just kind of to showcase that we can have very high scale and overall numbers to really subcellular and very, very fine detail. And I think often it's a problem that when people come to us, that they're not quite sure what they really want to analyze. And sometimes it really only comes out during the analysis and say, oh, have you thought about this? Or have you looked at this? Or this would make sense to look at, right? So there's no kind of right or wrong answer. I just think it's always important to reconsider that in theory we could analyze everything, but sometimes it just makes sense to look at very particular or very specific sub points really. And so with that, I just want to very briefly summarize that I've now spoken a lot about, zebrafish, imaging and understanding, pre-processing and segmentation and quantification. But I think the most important thing is that you have to have this conversation with different scientists. So from a biologist to a microscopist to a data analyst, I think all of us have such important roles to get the best data and the best research done. And we can learn so much from each other. And I think today I just wanted to show you, a little bit kind of from our perspective, from the data analysis, what we can do. But I think that interaction and exchange is the main thing for all of this, really. And so with that, I want to thank a lot of people. So today's data that I showed were from my PhD at the University of Sheffield with Paul and Tim, as well as from UCL with AiryScan data with Ryan MacDonald. I also want to thank all the students, collaborators, communities, and all the funders who obviously have really contributed to all the work that I've been showing today. So it's a lot of people, that are involved in all of these projects. And so a huge thanks to them, but also a huge thanks to everyone and in all the audience. And I'm happy to take any questions.
Thank you, Elisabeth. It was really great. The floor is open for any questions. Please write it into the chat box or just raise your hand here via Zoom and I will look out for it. During the talk, there was Thomas Nadera who raised a hand, but I think he's not anymore online. So while people are thinking about questions, I might ask the first one. So you have chosen a lot of, the nice example with the zebrafish and vessel-like structures. What do you think, how far can you employ the segmentation technologies, for example, the shape-specific filters if you look at more fine-tuned tube-like structures or filament-like structures, I'm now thinking about cytoskeleton or mitochondria tube-like structures. Is it really like we could apply basically the same on the more finer structures?
Yeah, that's a really good question. And actually, so yes, you can, is the quick answer. And the long answer is the main thing is to find the right scale of the filters, because these filters need to have an assumption about how large the vessels are. So, for example, if you would have very small and very large vessels, that could be a problem because the filter is normally applied in one size. So if you would, for example, think mitochondria, they're normally always more or less the same thickness, right? So that would be fine. So that would be great. So you can measure a few mitochondria and say, okay, my average is, for example, let's say 3 microns. I don't know, sorry. But if you say that more or less, then you could apply the filter at about that size, and that would be fine. But obviously you could fine tune that and say, okay, what does it mean for my data? Can you, make the filter a little bit larger, or a little bit slower, or like lower? So yes, definitely there is scope for that for anything that's a line or tube-like structure.
Okay, perfect. There's another question from Pete Curry. Steve, I think you allowed Pete to talk.
Yes. Can you hear me?
Yeah.
Oh, that's excellent. Thank you for a great talk. One of the things that I find equally bewildering as the amount of data collected in different methodologies is the amount of segmentation and analysis tools that are presented. I'm wondering, is there a clearinghouse or a customer satisfaction survey or some rating for some of these segmentation programs? Because as fast as we're producing the data, our colleagues in the computational world are producing excellent tools to use. And we would love to know which ones work the best and where to find out about them, because mostly we just search the literature and we pick one that seems to work, but that seems to be a very uneducated way to do it. Is there a better resource-driven way that we can evaluate and advocate for tools that are better than others?
I love that question. Thank you. Yes, I've spent so much time on segmentation in my PhD. I've got nightmares. No, it's great. So the thing, like you say, is there's so many different tools, and that makes it obviously really difficult. I would always say, like, stick at the start with something that you're familiar with. So something like a general filter or, a very simple segmentation, and work your way up from that. What we found is comparing different segmentations on the same data is actually the best way for us. There's unfortunately no right or wrong answer, right? And there's very little work actually on going on and validating your segmentation, right? How do you actually test that it's really working well? And so the main problem that I find is that a segmentation works really well for one person, and then I get the same data from a different person and it doesn't, because there's such a huge variability, right? And so that's something that, again, there might be some fine-tuning that is needed, etc. But I think, so what we found is, especially with this tubular filtering, it standardizes the data so well that the segmentation is kind of secondary to it, if that makes sense. But yeah, unfortunately, there's no right or wrong answer. And the main thing is really testing different methods on your data and finding what works the best. So for example, this exsanguination data, or you can test how robust is it against noise, which is something that we also did. So you can either lower your laser power, so you acquire something with high, medium, and low laser power, and try how stable your segmentation or your process is. Or you can, for example, add artificial noise, which is what we do in machine learning a lot. So that's another approach. Or for example, you could take something where you, again, know that there's a difference, such as 2 different transgenics, and see how that compares. So it's unfortunately, not a straightforward process, and it takes a lot of like testing and looking at how does it work. And I'm sorry, it's a lot of work, but that's the thing that we really spend a lot of time with to get it right, because it is just so varied. And as you can imagine, a blood vessel is completely different to, for example, a macrophage or a microtubule, and so there's no right answer that you can apply to everything, and each data set will be very different. So,
I just wondered if there was a simple list of programming tools and previous applications that could somehow, because we have lots of repositories in Zebrafish like ZFin or somewhere like that. Could there be a digital archive of tools that have been used in Zebrafish and what for? because, other than trawling through the literature endlessly, and for a non-computational person, it's often sometimes hard to pick a tool and give it a try. That was just really one observation.
That's a really good point. I don't think that there is like a repository. I mean, they're very good reviews. They give you like an overview about, the different approaches that could work, so to speak. But yeah, I can't think of anything. If I can think, I will get back to you, but I can't.
That's all right.
Right now. Yeah.
Jenny, I think the chat's actually disabled, so I think I've only got sticking up their hands.
Thanks for letting me know. Okay, if anyone has questions, just raise your hand, and that seems to work. I don't know, Steve, if you can fix that somehow. But if anyone has a question, just raise your hand then, please. Gian, there. Okay, there's a question in the chat box now from Gian. I will read it out, Elisabeth. So Jihan says, you mentioned, thanks for a great talk. You mentioned people presenting you with subpar data sets. How do you address that?
Yes, often. So it depends what the problem is. Like I say, especially if it's sampling frequency, that's unfortunately something that I often ask people to go back on the microscope, especially if there's a huge problem, such as, for example, like the one that I showed. If people ask me to show these fine cellular protrusions, but actually they image it in such an interval that the microscope can't pick up on these, then I can't do something with it. So if we can't see it in the data, then I can't analyze it. That's basically as simple as it is, right? Another thing is often, for example, noise or low signal. And that's more something that we can work with, as in like pre-processing and reducing background and things like that. So I will definitely always give a shot on like doing the best that we can. But if there's a huge problem, like I say, it's this sampling frequency, then, we're all kind of, at a certain limit. But I think, yes, the main thing for me is always spend a lot of time with the data to understand what are the problems, what can I address, what does the person on the microscope need to address, and what is the thing that the biologist could maybe address, such as, for example, the staining. So I would really say this pre-processing and understanding what is the problems is the main thing to getting a good and solid data analysis going.
Hey. Great. Now, I think it was my mistake. So please write your question into the question and answer box and not the chat box. So question and answers are coming through. Azadeh has also a comment here in the question and answer. She says, Elastic software showed a better segmentation for cells according to the current imaging conference in Monash. It's more a comment. I don't know if you have a comment for that as well. What do you think about the Elastic software?
I think it always depends on the data. Unfortunately, there's no right or wrong answer. I think that generally machine learning will be kind of the way forward. But again, I am a biologist by training, so I understand, people are being scared of code, and I appreciate that, especially machine learning is something that is, especially in the next few years, probably not yet common in all the labs, maybe in the future. But for example, I even think, having to install Python or Elastic can be a huge problem for a lot of labs. So I personally usually try and still work with Fiji and ImageJ, because that's what people are the least scared of, and they are more likely to use that and to work with. We can now integrate some of, for example, Python into Fiji, which is great. But I'm trying to do that in the background, so people don't have to, work with Elastic or Cell Profile or anything that is, more of a hurdle. Because I think that is also a problem, that a lot of people they get something that they, some code, and they could run some analysis, but they're too scared of it, and then they never use it, and they say, oh, I go back to counting my cells, or, oh, I go back to, just measuring it because it's easier and I think it's quicker. And so I personally have seen it a lot of times, and so for me, yes, Elastic is great, but it's definitely, again, a use case question of what you're looking at. And I'm personally a huge fan of Fiji and ImageJ, just because it's so, simple or easy to use for lots of people.
That's great. That's, I think, a very good point. It's not only like what you can do, but also how user-friendly it is indeed. Yeah. Okay. Are there any more questions? Please raise your hand or use the question answer box. I think we have all them. So I can't see any more questions. With that, yeah, Elisabeth, I thank you very much for a very nice presentation and taking the time early in the morning to present to us here in Australia. And yeah, thank you very much.
Thank you so much, and thank you for everyone for joining and all the great questions. Thank you, really appreciate that.
No worries.